So when I did this talk for my master's, I wore a frog onesie. Unfortunately, I can't find it. So just imagine I'm wearing it and keep that in the back of your head. So I'm going to do it a little opposite direction. So I'm going to start with my acknowledgments. Um, if it weren't for these people, I wouldn't be here right now. So I feel it's best to give them, credit before I start talking about my nonsense. So these people helped me get me where I am now, early in my career. They gave me opportunities that I don't think I deserved as a, you know, rebellious, very annoying kid, I'm sure. So I'm very, very grateful for them. I also want to give a shout out to the high school teacher that told me I was an idiot. So look at me go. And also, I'd like to thank my grandfather. His passion and everything he just used to love so much about amphibians is what got me so curious about them as well. So thank you also to everyone who helped me in the field. Um, One of them is actually in here now. So without them, I would not have the data. And yeah, they would put hours in, sweating and not complaining. So yeah, really, really, really lucky to have them help me. Um, Got funding from a few places, North Carolina Herpetological Society, Study of Amphibians Reptiles, and Robin got funding from Illinois Department of Natural Resources. So I appreciate their money. That said, thanks to my advisors, Robin and Jason, really helping me get along here and get towards the end of the light. So appreciate your help. Thanks to my committee members. I found your pictures online. All right. Thanks to fellow graduate students. It's been a long 5 years, but it's been fun. Uh, Friday Journal Club, shout out. I'd like to thank my mom who's here today, my brother, partner Emily, and our children, Bugbert and Fig Newt. I'm doing it for them. All right, so my broad dissertation stuff will be broken into three major questions, chapters, if you will. So the first one is studying tree frogs that don't want to be found. Second, studying a salamander that doesn't want to be found. And then third, studying larval amphibians afraid of being eaten. But before we get there, we got to take a step back. We got to think holistically. So let's think about ecosystem function in general. So ecosystems are complex networks of biotic and abiotic processes that provide essential global services like nutrient cycling and storage. Proper ecosystems depend on specific groups of organisms within specific habitats that enable them to persist, grow, and reproduce. However, habitat availability and condition are especially important for organisms with complex biphasic life histories, which undergo substantial developmental and habitat shifts. Example being those that transition from aquatic larvae to terrestrial adults. So amphibians are one such group, and they play a very crucial role in these processes at the ecosystem level. It's mostly because of their unique life history, which depends on both aquatic and terrestrial habitats. So typical reproductive and developmental stages of this aquatic amphibian: lay eggs in water, they hatch, that tadpole then metamorphoses a miniature version of the adult, grows, and then lay eggs, the cycle keeps on going. Same with salamanders in our region too. Same general principle. I also share in that I also went through metamorphosis to get to where I am today. All right, so not only are amphibians obviously very cool, they're also really important. That's how important. All right, so amphibian life stage specific behaviors are critical for ecosystem function. These are talking about linking aquatic and terrestrial food and nutrient webs. Amphibians are also the primary regulators of invertebrate populations. They're consumed by numerous other organisms, and they can be used to indicate overall ecosystem health. Sadly, many amphibian populations are declining. In fact, amphibians are one of the most threatened vertebrate groups, with about 41% of the species facing extinction. An example being this Wyoming toad here. There are about 2,000 individuals left in the wild. But why is that? One possible reason is I try and lick every single frog and salamander I see, but there could be other reasons. So disease. So we have chytrid fungus and ranavirus are the large disease types for amphibians, but also we have habitat disturbances from mining, agriculture, and urban development, which can entirely decimate watersheds and ecosystem scale things. From that, we can have rundown of disturbances occurring like toxicity, in this case, from mining drainage. But also we have shifts in local rainfall and temperature patterns. So this is climate change, where something like, say, a wetland that normally holds water for, say, 90 days, maybe 1 year, that pond dries out really early, and that can have really catastrophic consequences for population viability in the long run. So the system that I'm going to be talking about is a swamp wetland ecosystem. I'll be using swamp and wetland somewhat interchangeably, but swamp is just a specific type of wetland found in southern Illinois, or the region that my research was in. So wetlands in general rely on interconnected aquatic and terrestrial processes, much like amphibians. They are biodiversity hotspots with complex species interactions, which provide flood mitigation, carbon storage, and freshwater resources. However, this dynamic nature also makes them highly vulnerable to environmental changes, which can also make them very difficult to study. So the habitat itself can be difficult to study, as well as the amphibians in them. But why is that? So amphibians are really cryptic, typically small-bodied, and often nocturnal. Also, their detection is complicated by life stage-specific behaviors, complex and irregular habitats, environmental variability, and imperfect sampling methods. So studying their population dynamics across life stages requires refining both survey methods and analytical tools. So I asked myself, what questions are worth spending 5 years of my life asking about animals that don't want me to find them in the first place? So I thought, let's try this. How do survey and environmental factors shape amphibian population dynamics in a bottomland swamp wetland? Sound like a good idea? I don't know. Maybe it is. We'll find out. But first, we need a place to do this, right? So this is Buttonland Swamp. Buttonland Swamp is a relatively small remnant bottomland cypress tupelo swamp wetland in southern Illinois. It consists of second growth forest with emergent permanent wetlands and ephemeral ponds interspersed around the Cache River. So what can we do with 5 years worth of Buttonland research, and what questions should we ask? So the first question that I decided to ask is, how can we accurately determine the habitat features and environmental conditions that drive post-metamorphic non-breeding tree frog presence and detection in terrestrial wetland habitat when they don't want to be found in the first place? So this brings us to chapter 1, Lord of the Trees, the Fellowship of the Tree. No, I'm just kidding. It's factors influencing occupancy and detection of hyalid tree frogs within artificial PVC refugia. This research was published earlier this year in Herpetologica, and it's still actually ongoing. There's plenty of stuff, still more to come out. So when we're talking about studying tree frogs specifically, it's pretty difficult to study their complete year-round population dynamics. Most previous research is really focused on the larvae, and that's because they're aquatic and they're easy to capture either by depth netting or trapping. But they're not always going to be aquatic. Eventually, they're going to metamorphose and they're going to have to leave the water. So how do we study those post-metamorphic animals? Because don't they live in trees, as the name suggests? Well, the easiest time typically to actually detect them is while they're breeding. This is because the males will aggregate around ponds and vocalize, which will draw in females. So we can literally just listen and record it and know when and where they were breeding. But what if we don't want to disturb their breeding habitats and activities because maybe there's a state-threatened species present, like there is at Buttonland Swamp? So since we know where they are when they're breeding, because we can hear them, Maybe instead we should focus on where they are when they're not breeding. That sounds right, like easy enough, right? So what could be the problem? Well, when they're not breeding, they're like really hard to find, actually. Can anyone find the frog? No, you can't. There it is. So they're really cryptic. And when they're not breeding or hiding, they're actually probably on the move. And they like trees a lot. And they're pretty much always in the trees. So how can we study a post-metamorphic tree frog population when they're hard to find most of the time? Well, we could maybe give them beer. These frogs seem to like it. That one's having fun. Or instead, what if we could give them something that resembles something that they would already normally use? In this case, what if we gave them a fake refugia that we can put on the side of a tree that maybe they'll utilize? So artificial PVC refugia is the solution to this. Multiple studies across the planet have found that various tree frogs will use these as refuge from environmental and predatory pressures, very likely similarly provided by natural tree hollows. So this can allow for us to potentially capture them year round. Also, PVC pipes are really inexpensive. These traps are easy to make, and they're very durable, so they can last a long time. And they're capped with the bottom, which allows for retention of water, which can be very helpful in really warm conditions where they want to conserve moisture. So what I did is in the fall of 2021, I put 70 PVC refugia on trees within the aquatic terrestrial ecotone of the largest pond in Buttonland swamp. So we can see here underneath all the colors, that's basically the contour of the pond. And I put all the pipes right where that water would eventually go at its highest peak during the breeding season. So there's 7 rows of 10 pipes, more or less, distributed equally. So we check the pipes twice a month for a year. And then whenever we found an individual in a pipe, we would mark it with a visible implant elastomer, which looks like that. And then we would measure them, age them, and sex them. So by marking them, we were able to keep track of individuals as they grew. So an individual that started out, say, a juvenile, when it grew, we could update that individual's age to an adult so we can get essentially really, really dynamic data on them. So with this information, I wrote some Bayesian models to estimate the strength, direction of the habitat and survey influences on their occupancy and detection within these refugia. So the two things we're looking at, occupancy and detection. Occupancy is the probability of a refugium being occupied by a tree frog. There are a lot of different potential covariates and parameters that could influence this, but I'm just going to cover a few that we thought would be important. So this would be canopy cover above the refugia, cardinal direction of the refugia, height on the tree, and the ratio of previous surveys the refugium was occupied. And then for detection, we have the probability of detecting a frog in a refugium, given the fact that the frog is occupying the site or the refugium. For this, we have number of days since the last rain, which we thought might influence them being detected, as well as the survey date and survey temperature. So using some basic occupancy models from McKinsey, separately modeled the whole community and each distinct age sex group, which would be subadult females, subadult males, adult females, and adult males. And then built models with non-correlated parameters, iteratively chosen by model selection. For this first set, I used uninformed priors because we didn't have a whole lot of information about the animals in this region and what they might be doing with these pipes. Fit the models in R with JAGs and report only the top models for the community and each age sex group. So here's the basic model structure. The thing that you should only really focus on is the fact that detection is conditional on occupancy, which makes our estimates much more robust and it makes it more realistic. So from this, we got 179 tree frog detections and 42 of our pipes. Not bad. And we detected 3 species. Hylovoca, which is the bird voice tree frog, which is the Illinois state threatened species, green tree frog, Hyla cernaria, and then Hyla chrysosceles, Cope's gray tree frog. Almost all the captures were Hyla chrysosceles, so that's what we used for models. Looking at refugia fidelity, so essentially if a pipe was occupied, there's a 77% chance it was going to be occupied in the next immediate survey. If a pipe was occupied previously and it wasn't occupied during a certain survey, there's a 46% chance it would be occupied during the next survey. So frogs typically return to the pipes and use them pretty consistently. Importantly, we found no difference between the fidelity rates among the age sex groups, which is important as far as assumptions are concerned about not having any factors related to the age groups overlapping. So looking at our community occupancy probability, we have an actual estimate, and this says on average, an individual PVC refugium has about 80% probability of being occupied by at least one tree frog at any given time. We then split this up into the separate age sex groups, and we can see there's relatively similar estimates. So these broad dark bands are 95% credible intervals, and them overlapping indicates that there's no distinct variation between the means and their posterior values, essentially. So there's no difference between these groups, but there is some, it looks like there's at least variability visually. Now we're looking at detection. Given a tree frog is present in the PVC pipe, we have about a 25% chance of detecting it during a single survey. So our detection probability as humans is pretty low. And when we're looking at the age sex groups, we actually do see some true differentiation. In this case, subadult females are less likely to detect, or we're less likely to detect subadult females than the other age groups, but there were no differences among the other age groups. So recall these characteristics that we thought might be having an effect on occupancy. When we modeled this, the most important one that came out for the community was the ratio of consecutive refugia occupied. So essentially, if a pipe's occupied, it's more likely to be occupied in the future, which makes sense. Among the age sex groups, we see the same trend for this consecutive occupancy trend among all the age sex groups. However, when we look at some of the other characteristics, we can see that there's variability in certain things like cardinal direction or refugia height, canopy cover. This indicates that these age sex groups might have different characteristics that they're preferentially either choosing or being forced based in some sort of fashion for their occupancy. And then for detection, we had those three main parameters. And the most important one for the community was survey temperature. Where temperature increases, so does the detection probability. Interestingly, only 2 age sex groups had survey temperature as important aspects of their models. But adult males, interestingly, had days since last rain, and it was a pretty strong relationship where there was about a 30-day drought period, and we had the highest detection probability of them. And for Julian date, we have this trend where subadults were more detectable early in the year, followed by adults later in the year. And this is mostly because those subadults, we were tracking them, and as they grew, they were updated to being adults. So that makes sense. So to summarize the fellowship of the tree, the age sex groups had similar probabilities of occupying the refugia. If a refugium was previously occupied, there's a high probability of being occupied during the next survey. Subadults were primarily detected early in the year, while the adults were detected later in the year. Subadult females were least likely to be detected, suggesting either alternative refuge use or also potentially a lower rate of female recruitment in the previous year. So survey temperature and days since last rain positively influenced detection and community for some of the age sex groups, indicating some of the tree frogs might have been preferentially utilizing the pipes as actual refugia from climatic stressors. So overall, this study validates the use of PVC refugia to gather nearly year-round population level data on post-metamorphic tree frogs in our Southern Illinois bottomland swamp wetland complex. So what comes after this? So I'm working on these right now. Parts 2 and 3, the 3 tree dwellers, the 2 towers, I don't know, whatever you want to do. And then the return of the tree. So what I did is we continued the study for 2 more years, and we got very, very high resolution data on a bunch of individuals, on their detection, growth, movement, for all 3 species and their distinct age sex groups. So with this data, I am currently working on manuscripts that will examine how temporal climatic shifts influence the species population responses, such as survival, recruitment, and then how these shifts potentially affect their growth and movement patterns. So why should we study animal movement? So studying animal movement is key to understanding various population dynamics. This data can be used to estimate home ranges and identify key habitat for important life stages and natural history behaviors, such as reproduction, foraging, and refuge use. This information can directly inform conservation strategies and provide otherwise inaccessible insight into critical ecological processes. However, most of this research has primarily focused on large, conspicuous organisms, which leaves a pretty big knowledge gap for smaller and cryptic organisms like amphibians. So for the second question, I'm looking at this interesting salamander that does not want to be found. How can we identify and track amphibians without having to physically recapture them like we did with the tree frogs? Given that, how do we then determine if the marking and tracking method is working correctly? But before all that, how can we objectively capture the amphibians in the first place within a heterogeneous, irregularly shaped, and variably sized ponds within Buttonland Swamp? So chapter 2 paper published somewhat recently, Animal Biotelemetry, a novel indicator to assess pit tag retention in an aquatic amphibian. So why is studying amphibian movement difficult? Well, the tree frogs, we could mark them with a VIE, right? We could keep track of them as we recaptured them in the pipes. So it's pretty convenient. They come back to the pipes and we know which one it is. However, most amphibians don't do this. They're not very detectable or catchable to begin with, and they don't really use refugia, and some of them are really bad at using traps. So physically recapturing a VIE marked amphibian is very, very unlikely. So what do we do then for amphibians that don't utilize these things, such as tree frogs, and ones where traditional tracking methods like GPS and radio telemetry aren't really going to work, either because the amphibians are too small or because of their fully or semi-aquatic natural histories? A passive integrative transponder tag. So this allows us to keep track of an individual. It is a subcutaneous implanted microchip that requires no internal power, and it can allow for the indefinite, rapid, and accurate identification of a previously tagged individual. They can also be pretty small. So most of these tags can be detected up to 34 centimeters away through water and bedrock by a transceiver when activated by an electromagnetic field produced by a portable antenna like this. So with this antenna, we can actually do active scanning surveys, which would allow us to detect and ID individuals and get their GPS location without the need to physically recapture them. This could be really, really helpful for cryptic and invasive organisms such as amphibians, especially if they're aquatic. So a review looked at what we should consider the size requirements for individuals based on the tag. They said that there should be about 17.5% the size of the body length, or not exceed 7.5% of the body length. So for the largest pit tag, 32 millimeters, that would give us a minimum body length of 185 millimeters. The industry standard current pit tag is a 12 millimeter, but with that, we would need the animal to be at least 69 millimeters. Our tree frogs were on average 36 millimeters long, so they're obviously way too small for those tags. That's where insert the 8 millimeter pit tag. In the late 2010s, Biomark introduced this micro 8 millimeter pit tag, which is shorter, smaller, and lighter than the previously smaller 12 millimeter tag. So when we look at the size recommendation for this, the minimum body length should be 46. However, that's still way too big for these frogs. So obviously, we're going to need a bigger amphibian. And what about this thing? It seems like a good fit. The lesser siren, Siren intermedia. It's a very strange salamander. They inhabit permanent and ephemeral ponds throughout the eastern United States. They are neotenic or netonic, where the adults retain their external larval gills and an aquatic-ish life history. Also, they only have limbs on the front of their body, which is rather unique. They are just one of two salamanders known to vocalize, yet we still don't know exactly why. They are also the only salamander known to have adapted to an omnivorous diet, which is absolutely insane. They are capable of both avoiding and escaping common amphibian traps. They're so smart, in fact, they can almost spell their own name. They also do this weird thing where they can estivate in underground burrows during periods of drought. They do this by enveloping themselves in a mucus-like cocoon, where they can remain for over a year. And they're also one of the largest salamanders in the world, reaching over 2 feet in length. So they're obviously an ideal species for pit telemetry. So previous studies have actually looked at using pit tags and siren. They would exclusively use the large 12-meter tags, and they would only do it to identify recaptured individuals from nets or traps. Previous studies also showed that the juveniles can't retain these large tags as the surrounding tissue rejects them and then would eventually eject them. So our knowledge of juvenile populations is pretty limited. Interestingly, no studies have actually considered using the 8mm pit tag in juveniles. Also interesting, no studies have even assessed the general adult or juvenile siren movement and estivation patterns using the pit telemetry. So this novel 8mm pit tag has been used by a few researchers in a few different amphibians. However, none of them have examined the factors influencing their use and retention. So using juvenile siren intermedia, we aim to determine the animal size and marking parameters that influence successful retention. We then assessed how to use a tag's telemetry detection history to determine if it had been retained. But first, to do all this, we have to catch them. But like I said, they're very evasive and they're adept at escaping traps. However, we could potentially mitigate this by using dip nets, which they seem to not be able to escape. So previous research has found or designed studies to do this area-scaled effort for the dip net surveys. Typically, this was designed with ponds that are relatively similar in size, shapes, and microhabitats. However, Buttonland Swamp does not have this. All the ponds are very different, which makes it difficult to consistently sample them using the traditional area scaling method. So our preliminary analyses, the surveys found that we find individuals about 4 meters from the shore and never beyond 10, which is likely because of the absence of dense leaf litter that they use for cover. So with that in mind, developed this perimeter-based method. So we modified the area scaled method to conduct the dip net sweeps every 10 meters along the pond's perimeter. So perimeter scaling actually optimizes the standardized survey effort and habitat closer to the shore in irregular shaped ponds better than area scaling would. So given this, at each sampling location, we perform 3 vertical sweeps, each 2 meters forward. By doing this, it minimizes capture biases while maximizing detection within that first 10 meters from the shore. This also allows for randomized sampling in key microhabitat, which is most likely occupied within that 10-meter zone. So for 2 years, we conducted dip net and trapping with funnel and trash can surveys every 15 days in 2 ponds that were known to have siren. Whenever we captured a juvenile, we would measure it and then inject it with an 8mm pit tag posterior to their cloaca, their butthole, and then we would record their GPS location. So to assess whether the siren's size or the pit tagger's overall confidence influenced tag retention, we recorded the total length of the siren, the tail length, which was probably thought to be most important because it's the area physically being tagged, and mass. And then to get at the confidence, we recorded Julian date, thinking that the tagger's confidence would increase over time as they got more and more practice. So to get some of this data, we would do these active pit telemetry surveys. We did these twice a month, where we take the scanner and go across the maximum extent known wet perimeter of the pond, scanning randomly for 45 minutes. Whenever a tag was detected, the survey recorded if the inundation state, so if it was wet or dry, and then flagged the location. So that way we can come back during the next survey and scan and see if it was still there. If it was moved, then we remove the flag, and then we wouldn't preferentially survey that area the next time. With this information, we then calculated the straight line distance between each subsequent detection. So with this information, we were going to try and figure out, can we figure out which retention category each tag belongs to based on the telemetry distance information we have now. So Biomark is the company that makes this product, and they report their own accuracy. However, we observed a little bit of a different value. So they report 3 meters within the reader's location. We found the accuracy more or less is closer to 5. We had known drop tags that did not move, and on average, they were about 5 meters apart in error. So based on this, we can assume that the GPS error for subsequent dropped and non-moving pit tag telemetry detections in our study area with these 8mm pit tags is less than or equal to 5 meters. Just making sure you're still paying attention. I think that's a salamander. All right, so with this, we can now determine and assign each pit tag to a retention category. So drop tags have to have 2 things. They have to have at least 2 telemetry detections with a redetection average distance less than 5, which is the average distance between telemetry redetections. We can see this here. First captures the dip net, and then it was scanned 4 times. So we'll take the distance, the average of the distance between each scan, and if that value is less than 5, it's strong evidence that that tag was dropped. It also has to meet the second characteristic, which is the second last, being less than 5, which is the distance between the second and the last detection. So using that same example, we can see the first scan and the last scan, and that distance is also less than 5 meters, giving us strong evidence to suggest that tag is indeed dropped. So for retained tags, they have to have one of these 3 things. They can either have the redetection average distance and second last values greater than 5. They could have been only telemetry detected once and then never seen again, or they could have been captured and scanned from a physical animal and then never telemetry detected. So with this in mind, we developed 2 sets of models to assess first tag retention and then second telemetry movement and detection metrics. So in the first model set, we did pit tag retention. We used generalized linear mixed models to assess the relationships among pit tag retention and initial size and marking parameters. So these include tail length, total length, mass, and Julian date. In our second model set, we were looking at size and telemetry movement metrics. These linear models examine the relationships for the initial size marking parameters to various movement metrics. There are a lot of movement metrics, but I'll only include 4 here. First distance, which is the first distance moved. Second last, average distance between all detections, and then redetection distance. So from this, over thousands of dip nets and trapping attempts, we caught a whopping 67 animals. Siren do not like being captured. And almost all of them were found in dip nets. 3 animals were caught in funnel traps and none in trash cans. Of those, we redetected 20 of the pit tags. 2 of those were in individuals we physically recaptured, and the remaining were ones that we detected during our telemetry surveys. From those values that we calculated earlier, we determined that 11 juveniles retained their pit tags. So in the first model set, The top model indicated that tail length was the best predictor of tag retention. So as tail length increases, the probability of that tag being retained also increases quite rapidly. Which makes sense. There's more room, there's more tissue area for that tag to be received, and it's less likely to be ejected. All right, so the second model set, our top model, indicated tail length is a great predictor, and the movement metric was average subsequent redetection distance. So all secondary detections. Only 3 of the 11 animals had multiple telemetry redetections. However, even with that small number, there's clearly a very, very strong trend here, where as tail length increases, the distance between those redetections also increases. In the second supported model, less supported, but still pretty strongly supported, again, we have tail length, and then we have all redetections. So here we can still see that same general pattern, whereas tail length increases, so does the average distance that those animals were detected moving. So basically this shows us the tags were most likely expelled from juveniles with tail lengths less than 50 millimeters. Based on that predicted threshold value, this is actually 9% longer. So this really kind of shows the importance of establishing size requirements for effective telemetry applications. This is really important, though, when there's different age classes that require potentially different size tags. Also, this perimeter modified dipnet survey method that we came up with can clearly supplement or even replace aquatic amphibian traps. Although it is definitely much more time intensive, it could potentially address biases in traditional trapping in these heterogeneous habitats. So additional paper coming from this is looking at the individual movements of those individuals that did retain their tags. So we also did pit tag adults with the larger 12-minute tags. So this is just a simple illustration showing some of the movement patterns that we detected. All right, so far we've only asked questions about post-metamorphic amphibians. But what about their aquatic larvae? To recall from earlier, that disease, habitat alteration, and climate change are the primary drivers of amphibian declines. However, their vulnerability is compounded by their biphasic life history, as the larval stage is particularly sensitive to various abiotic and biotic factors. These include water temperature, DO content, pond hydroperiod, predation risk, and food and refuge resource competition. So not only can these influence survival for the larva to metamorphosis, but it can also impact subsequent adult health, reproductive success, and population viability. So this brings us to the last chapter question. By modifying the model framework from chapter 1 and using the perimeter scaled dipnet methods from chapter 2, can we determine the habitat and climatic conditions shaping the population dynamics of a diverse larval amphibian community and its species across Buttonland Swamp? So this is the paper that came from this. It is currently in review at Freshwater Biology. Let's hope for the best. But it's the key habitat and predatory influences on the community and species level population dynamics of spring breeding amphibian larvae within a remnant tubulo-cypress wetland. I think we could probably shorten it down a little bit. Pretty long. All right, so in the spring of 2023, Buttonland Swamp was not doing well. Local rainfall patterns limited to only have 4 water-filled ponds with defined boundaries. Luckily, however, the ponds varied in various parameters like size, hydroperiod, depth, predators, and canopy cover. This variability made it possible to examine which specific characteristics might be strongly influencing the larval amphibian populations. However, if you can count, there are obviously some potential limitations and considerations with this. So based on Buttonland Swamp and what we had available, we clearly did not have a very large sample size, nor did we have any pond type replication. So with this in mind, we instead focused on improving our precision and interpretability of our estimates by doing a few things. First, maximizing our pond standardized survey effort using that perimeter-based method. Maximizing temporal survey replication. Incorporating community and species level models into the same framework. And utilizing robust hierarchical modeling approaches. So for what we did for the actual study, it was over a 6-week period in spring of 2023, where we conducted 3 systematic dipnet surveys at each pond at 14-day intervals. This time period was close enough that we can assume closure to migration, and death. So we use the same dipnet methods from chapter 2. And after each sweep, we would put the larvae, inverts, and fish in separate containers so they wouldn't eat each other. When we were done with all that, we would then identify and count each of them. So there are a bunch of things that we measured, but I'll only talk about a few of them. So perimeter, canopy cover, leaf litter, vegetation, depth, dissolved oxygen, temp, dipnet effort. Interestingly, dipnet effort was not correlated with parameter. We also did predatory fish presence, the number of back swimmers, which are these things if you're unfamiliar, and then also crayfish, beetles, and dragonflies, all of which are known predators of aquatic amphibians. So using a hierarchical modeling approach, use this multi-species abundance modeling framework, which allows us to estimate both community and species level relative abundances and occupancy detection probabilities within the same model. By linking multiple species into a single encompassing hierarchical model, it improves the precision of the community and species responses to environmental and survey covariates, as rare species get to borrow strength from more common ones. So these multi-species abundance models, they separate submodels for occupancy, abundance, and detection that are still separate but linked in certain regards. We also can improve our ability to estimate certain parameters by including random effects for site, species, and survey. This model structure ensures robust inferences despite environmental variability and a low sample size. So we built numerous models using combinations of non-correlated parameters chosen by model selection. For this model, we did use slightly informative priors. By informative, I mean if we knew a certain thing was likely to cause a certain result, then we could give it either a positive or a negative value. For example, hydro period, we know hydro period will have a negative effect on larvae because if it dries, they die, right? So we can knowingly give it a negative value before we spit it into the model itself, which helps inform it better. So from all this, we report the results for the community and each separate species. So here's the submodel structures. You don't need to get too worried about it. Just know abundance, again, is conditional on occupancy, and detection is conditional on occupancy and abundance, which makes our estimates much more robust. Here is a sample of one model. That's half of it. That's the other half. I wrote dozens of these, so it was very fun. But don't get me started on like modeling code for plotting and checking. It's just, there's a lot of shit going on. All right, so from this, we got 516 dipnet sweeps, and we captured around 1,400 larvae from 6 species, 4 of which were salamanders and 2 were frogs. Quickly, there's some variability within the salamanders, where more or less 2 or 3 of them had the same-ish captures, and 1 was captured more often. Then we had 2 frog species. The chorus frog actually comprises 2 species, but we just linked them together because they're really hard to tell apart in the field. It appears that the frogs were actually captured more than the salamanders. All right, so when we do the modeling for the community estimates, on average, a given species has about a 70% probability of occupying a pond, which is pretty high. And then when we're looking at abundance, about 1,450 individuals were estimated to be present across all species and ponds. And then we're looking at detection probability. Given larvae are present in a pond, there is about a 25% chance of us detecting one during a single survey. If you recall from the first chapter, that's the same probability. It seems that the amphibians within this study area are similarly detectable. All right, when we're looking at the individual species for occupancy, we do not see any differentiation as all those credible intervals overlap. However, when we're looking at our abundance, we do see true differentiation where all of our salamanders do have lower abundances than the two frogs. And among those frogs, there is no difference, though it appears that the pseudacris or chorus frogs may be slightly more abundant. And then we're looking at detection probability. We see quite a bit of variability. However, there is no true differentiation between them, but there still indicates that they might be responding to various covariates differently. So when we're going to look at occupancy, abundance, and detection at the community level and the species. So for the first one, our top model picked three parameters. First one was predatory backswimmers. So as the number of predatory backswimmers increased, our community occupancy probability decreased. Among our individual species, we see a somewhat general trend. However, two species, a frog and a salamander, appear to have the strongest effect, a response to these backswimmers. Whereas these two salamanders appear to have almost no response, so they don't seem to care about these predators. The next important parameter was canopy cover. As canopy cover increased, occupancy probability also increased. We have a very similar trend across the species, but the strongest response was for mole salamanders, and the weakest was for our chorus frogs. Predatory fish presence, so -1 means no fish, and 1 means there are fish present. So in ponds with fish, occupancy was much lower. And we saw a very similar trend for all species when it came to predatory fish. So now we're looking at abundance. The first parameter in this was canopy cover. As canopy cover increased, abundance increased. Saw similar trends for the species, though spotted salamanders had the strongest response, while smallmouth salamanders had the weakest. This shows intra-order and intra-family variability within the same system. So these species are responding truly differently based on their own natural histories. The next thing was leaf litter, which as that increased, abundance also increased. And here we see some differentiation between our salamanders and our frogs. So the frogs actually have very weak responses to leaf litter, whereas the salamanders have very strong responses. And this reflects in real time from these models variability in their actual natural history behaviors. So it kind of validates the fact that this model is working. The last one is pond perimeter, and as that increased, community abundance also increased. And this was generally similar for all species. All right, so the last one, detection. For these, it was all predatory things that had a negative effect on detection. So predatory backswimmers had a negative effect on detection, which was generally similar across all species. Dragonfly larvae had a negative effect, which was also very similar across species. And then predatory fish had a pretty negative effect, which was more strongly affecting the frogs and one salamander. All right, so what does this tell us? Predatory invertebrates and fish suppressed detection, likely via avoidance behaviors, and species-specific tolerances varied, with salamanders showing greater resistance to fish. Also, survey effort had minimal impact, supporting the effectiveness of the perimeter method. Canopy cover, leaf litter, and perimeter were the strongest positive predictors of abundance, which indicates that structural complexity provides refuges and enhances species persistence. This also highlights the fact that robust hierarchical Bayesian multi-species abundance models can help improve our understanding of amphibian community dynamics and complex wetland landscapes. Ultimately, these studies are able to provide critical insights for amphibian conservation, particularly in the face of habitat loss and climate change. So we collected a shitload of data, and another paper that I will be working on is the summer version of what this was. I lost all my phone pictures when I got a new phone, so these are the only remaining ones from the field. But this is basically what a day in the life looks like. Trying not to get sunburnt and just having a good time. So with that, I will take questions if I have time.